Welcome back to the Deep Dive. Now, normally we take a topic and explore it for general interest, catching you up on the state of the world. But today
Today is different?
today is very different. We are shifting gears completely. We are entering coaching mode.
That's right. We're treating this session as an intensive, high-stakes tutorial.
I want every listener, especially you, to picture the scene right now. It is late at night. The rest of the house is asleep. You've got your third cup of coffee brewing black, no sugar, and you're sitting at a desk just covered in highlighters and sticky notes. You are an MD anesthesiology postgraduate student, and you are deep in prep for the Gujarat University exams.
And for anyone who hasn't been there, I mean, that is a uniquely high-pressure environment. The syllabus is just vast. The examiners are, well, they're seasoned, and they can smell fear.
They absolutely can. And to help you survive this, we haven't just pulled a few articles from the internet. I'm looking at the desk between us right now, and we have the heavy artillery.
We do.
We have the absolute Bible of the specialty, Mueller's Anesthesia, the 10th edition. We've got Stolting's Pharmacology, Morgan and Mikau, Guyton's Physiology for the basics, and for the cellular bedrock, we have Robbins' Basic Pathology.
That is a formidable stack, and we are going to need every single page because the topic we're covering today is, well, it's arguably one of the most complex, controversial, and high-yield topics in the entire curriculum.
Today, we are doing a deep dive into neuroapoptosis.
Neuroapoptosis. It's a word that I think strikes fear into the heart of exam candidates because it forces you to bridge two very difficult worlds. Deep molecular biology on one hand and ethical clinical practice on the other.
Right. So our mission today is simple. We're going to move beyond just helping you pass. We want to help you master the why, the how, and maybe most importantly, the so what of neuroapoptosis.
Exactly.
We want you to be ready to handle a short note question, a massive 20-mark long answer, or even those dreaded traps in the viva voce where the examiner just stares you down and asks, "Doctor, did you just kill those neurons?"
Yeah, precisely. That's the moment we're preparing for. We need to connect the dots between the molecular diagrams in Robbins and the real-time clinical decision-making in Miller. We are going to structure this logically, section by section, just like you would map out a theory answer.
Okay, let's get right into it. Part A, Introduction and Exam Framing.
Okay. Imagine you are sitting in the exam hall, or you're facing an external examiner. The topic of neuroapoptosis comes up. You need a strong opening. You absolutely cannot falter on the first sentence.
So where do we start? Do we jump in with the anesthetic drugs? That's our specialty.
Now that's the first trap. You must start with the pathology. You start with the fundamental definition, and you have to be precise.
How precise?
Don't just say cell death. If you say cell death, the examiner will immediately bounce and ask which kind. You need to use the language from Robbins. According to Robbins, apoptosis is a pathway of cell death that is induced by a tightly regulated suicide program.
A suicide program. That's a very specific, almost grim phrase to use in an exam.
It is, but it's the correct one because it implies agency. The cell plays an active role in its own demise. It is an energy-dependent process. The cell itself activates intrinsic enzymes that degrade its own DNA and its own proteins.
Okay, so it's an active process, not a passive one.
Exactly.
Now, almost inevitably, if you drop the word apoptosis, a classic Viva opener is asking for the difference between apoptosis and necrosis. This is, bread and butter stuff. But how do we distinguish them clearly enough to get those distinction marks?
You need to hit 3 or 4 key points of difference. These are straight out of Robbins and Guyton. You should literally visualize a table in your head for the examiner.
Okay, column 1.
Column 1, the cause. Necrosis is almost always pathologic. It's the result of let's say, a car crash at the cellular level, severe injury, toxins, ischemia. It's accidental, messy death.
Okay, messy and accidental. And apoptosis.
Apoptosis is often physiologic. It's part of the plan. It's essential for embryogenesis, for normal development.
Yeah.
It can be pathologic, which is what we're here to talk about, but its primary nature is regulatory and programmed.
Right. Okay, point 2, what about the morphology? If I'm looking under a microscope, what am I seeing?
This is a key visual. In necrosis, the cell loses its ability to regulate its volume. It can't control the pumps, so it swells up.
It gets bloated.
It gets bloated, and the membrane integrity is lost. It bursts.
Like a balloon popping.
Exactly. And what happens when a cell bursts? all those intracellular enzymes and contents, all that junk, it just leaks out into the surrounding tissue.
Which sounds messy.
It is messy. It alerts the immune system. So necrosis elicits inflammation. That is the key word. A huge inflammatory response.
Inflammation is the hallmark of necrosis. Got it.
Correct. Now contrast that with apoptosis. The cell doesn't swell. In fact, it does the opposite. It shrinks.
It shrinks.
Yes. The chromatin, the DNA in the nucleus, condenses into these dense little masses. And the membrane, this is crucial, the plasma membrane stays intact, but it forms these little bubbles. We call them blebs.
This is more of the controlled demolition. The building implodes on itself. It doesn't explode outwards.
Perfect analogy. That's exactly it. The cell then breaks itself down into these neat membrane-bound, bite-sized pieces called apoptotic bodies.
Okay.
And crucially, because the membrane stays intact until the very end, nothing leaks out. There is no inflammation. It is a silent death.
Silent death. I like that. That's a good phrase to remember. So we've defined the pathology, but why is this topic on an anesthesia exam syllabus? Why is an anesthesiology examiner asking me about cellular suicide?
That's the so what. If you don't answer this part, you fail the clinical application of the question. Miller and Morgan both highlight this because of a massive and, frankly, terrifying concern. The idea that the drugs we use every single day are general anesthetics might trigger this very process in the developing brain.
So the fear is that we, the anesthesiologists, are inducing this suicide program in healthy neurons when we anesthetize a baby.
Exactly. We might be taking a normal physiologic process and turning it into a widespread pathologic one.
Wow.
And this is obviously most relevant to pediatric anesthesia. Stolting discusses the FDA warnings on this at length, but it's also highly relevant in the ICU. Miller talks about apoptosis in the context of sepsis, ischemia, brain injury. It's everywhere.
In terms of exam patterns, then, how will this question actually appear on the paper or in the Viva?
You need to be ready for three main formats. 1, a short note, typically titled Apoptosis in the Developing Brain, or maybe Anesthetic Neurotoxicity.
Okay.
2, a subsection of a long question. It might be buried in a question on pharmacology of pediatric anesthesia. And 3, the Viva. This is often a trap. You'll be discussing something that seems simple, like safe anesthesia in neonates or risk communication with parents, and the examiner will pivot and hit you with this.
Okay, so we have the framing. We know the stakes are high. Let's go deep into the science. Part B. Basic cellular mechanisms.
This is very high yield. If you are writing a theory paper, this is where you're going to draw your diagrams and get your solid marks. You have to nail this part.
We said it's programmed cell death, but why? What is the physiological role of this in the central nervous system? Why would the brain ever want to kill its own cells? It seems counterintuitive.
Think of it like sculpting. Guyton and Miller both describe this beautifully. During embryogenesis and later synaptogenesis, the brain is a massive overachiever. It produces way more neurons and synaptic connections than it actually needs.
Like writing a rough draft of an essay with way too many words and then you have to edit it down.
Exactly that. Or like a sculptor starting with a huge, shapeless block of marble. You have to chip away the excess to reveal the final statue. Apoptosis is the process that removes the excess neurons.
But which ones? Is it random?
Not at all. It specifically removes the ones that haven't made proper functional connections. This pruning is absolutely essential. If you don't prune, the brain is just noise, not signal. It's inefficient.
That's a critical concept to get across. So death is actually necessary for a healthy, functional brain to develop.
Yes. Cell death is necessary for life.
Let's go back to the mechanics. You mentioned these neat little packages, the apoptotic bodies. Robbins mentions a specific biochemical signal that allows phagocytes to recognize these dying cells, the so-called eat me signal.
Yes, and this is a detail that examiners absolutely love because it shows you've read the text closely and you haven't just memorized a summary.
So tell us about that. What is the eat me signal?
It involves a specific phospholipid called phosphatidylserine.
Phosphatidylserine.
Okay.
Normally, the cell works very hard. It expends energy to keep this lipid on the inner leaflet of the cell membrane, so it's facing the inside of the cell, the cytoplasm.
So it's normally hidden from the outside world. Right. But during apoptosis, a group of enzymes called scrambloses are activated. And just like their name suggests, they scramble the membrane and they flip the phosphatidylserine to the outer leaflet.
So suddenly, this hidden molecule is now on the outside of the cell, waving a flag.
Exactly. It's a chemical flag that basically tells the macrophages, or in the brain, the microglia, hey, I am dying. I am packaging myself up neatly. Eat me now before I leak and cause inflammation.
That is a brilliant detail to drop in an exam. Phosphatidylserine flipping. It sounds technical and really impressive.
It is, and it's the molecular explanation for why there is no inflammation. The cleanup crew is summoned and arrives before any mess is made. It's an incredibly tidy process.
Let's move to the meat of the mechanism. Part C, molecular pathways of apoptosis. This is where I think a lot of students usually get tangled up, but you say this is exam gold.
It is, without a doubt. If you can draw this flowchart on your exam paper, you are already ahead of 90% of the candidates. You need to know that there are two main pathways, the intrinsic pathway and the extrinsic pathway.
Okay, let's break them down. Which one is the most relevant to us as anesthetists, the one we really need to focus on?
The intrinsic, which is also called the mitochondrial pathway. Miller explicitly links ischemia and our anesthetic agents to this specific pathway. So this is the one you absolutely must master.
Okay, walk us through the intrinsic pathway. Step by step, nice and slow.
It always starts with a trigger, and the trigger comes from inside the cell. It could be DNA damage from radiation, it could be oxidative stress, or, and this is the crucial one for anesthesia, the loss of survival signals.
Loss of survival signals. So the cell feels neglected or stressed. What happens next?
What happens next is a battle that takes place at the mitochondrial membrane. This balance is controlled by a family of proteins called the BCL2 family.
The BCL2 family.
Yes. And you can think of it like a tug-of-war. On one side, you have the anti-apoptotic proteins. These are the guardians, the good guys. The main ones you need to remember are BCL2 itself and BCL2-XL.
BCL2 and BCL-XL.
And what's their job? Their job is to keep the mitochondrial wall strong and impermeable. They are the off switches for the cellular suicide program. They say, don't do it.
Okay. And on the other side of the rope in this tug-of-war,
you have the pro-apoptotic proteins. These are the villains. The two you have to remember are Bax and BAC.
Bax and BAC.
Their job is to punch holes in the mitochondria. They are the on switches. They want the cell to die.
So it's BCL2 versus Bax, guardian versus executioner.
Correct. Under normal, healthy conditions, BCL2 keeps Bax and BAC in check. It neutralizes them. But when the cell is stressed, or importantly, when it loses those survival signals we mentioned, the balance shifts. Bax and BAC win the tug-of-war.
And what do they do when they win?
They oligomerize, which means they group together and they form channels or pores right in the outer membrane of the mitochondria.
The fortress wall has been breached.
Precisely. And this breach allows the release of a very, very important molecule that is usually locked safely inside the mitochondria, Cytochrome C.
Cytochrome C? I remember that from biochemistry, from the electron transport chain. It's supposed to be involved in making energy ATP.
It is. That's its day job. But when it leaks out of the mitochondria and into the cytoplasm, it takes on a new role. It becomes a messenger of death. This is often called the point of no return.
Okay, so once cytochrome C is out, that's it.
Pretty much. Once cytochrome C is in the cytoplasm, it finds and binds with a protein called APAF1.
APAF1.
Yes,
Yeah.
apoptotic protease activating factor 1. They come together, along with some other components, and assemble into a large wheel-like structure called the apoptosome.
The apoptosome, that sounds like some kind of sci-fi superweapon.
It effectively is. It's a weapon of cellular destruction. The entire purpose of the apoptosome is to find and activate the first of our key enzymes, Caspase 9. This is known as the Initiator caspase for the intrinsic pathway. It gets the ball rolling.
Okay, so that's the intrinsic path. Stress, BCL2 balance shifts, Bax bake win, holes in the mitochondria, cytochrome C leaks, forms the apoptosome, which activates caspase 9.
Perfect, you got it.
So briefly then, what about the extrinsic pathway, just so we can contrast it for the exam?
The extrinsic path is also called the death receptor pathway. And as the name implies, this is triggered from the outside of the cell.
Right.
A specific molecule, a ligand-like TNF tumor necrosis factor, from an immune cell, or another one called Fas ligand binds to a specific death receptor on the cell surface.
So it's like a lethal knock on the door.
It's a knock that killed you, yes. This receptor-ligand interaction on the outside causes a change on the inside. It recruits adapter proteins, which then directly activate a different initiator, caspase 8.
Okay, so the intrinsic pathway uses initiator caspase 9. The extrinsic pathway uses initiator caspase 8. Where do these two paths meet?
They converge on the executioners. There are several, but the main one you absolutely must remember for the exam, write this down, highlight it, is caspase 3.
Caspase 3. Why is it called the executioner?
Because Miller describes it as the final common effector. Once caspase 3 is activated, whether by caspase 9 or caspase 8, the game is over. It goes on a rampage.
What does it do?
It's a protease. It starts chopping up everything that's essential for the cell to live.
Yeah.
It cleaves DNA repair proteins like PARP so the cell can't fix itself. It destroys the cytoskeleton, which makes the cell shrink and bleb. It activates enzymes that chop up the DNA. It systematically turns the cell into those neat little apoptotic bodies we talked about.
And just to reiterate for the exam, Miller specifically links anesthetics to that intrinsic pathway, the mitochondrial leak.
Yes, that is the critical link you must make. The hypothesis is that anesthetics or ischemia lead to mitochondrial dysfunction, which causes the shift in the BCL2 balance, which leads to cytochrome C release, and then the whole caspase cascade. If you can draw that chain of events, you will get the marks.
Okay, we have the general mechanism down. Now let's get specific and look at Part C, neuroapoptosis. We were applying this whole process specifically to the brain.
Right. And Miller is very clear that neurons are a unique case. For one, they are postmitotic, they don't divide, so if you lose one, it's gone for good. And in the context of things like ischemia or drug exposure, neurons seem to be biologically primed to undergo this suicide program rather than necrosis. Although in a severe injury, like a massive stroke, you'll get a necrotic core with an apoptotic area around it.
So you can have both happening at once.
You can, but apoptosis is a key player in neuronal death.
And we mentioned this pruning role in development. Let's dig just a little deeper there. How does that work on a molecular level?
It's a beautiful system. As we said, the brain produces this excess of neurons. These neurons then project their axons out to find their targets. Think of millions of wires trying to find the right socket.
Okay.
If a neuron's axon successfully connects and forms a functional synapse, the target cell releases neurotrophic factors. The famous ones are BDNF, brain-derived neurotrophic factor, or NGF, nerve growth factor. These are the survival signals we talked about.
So the neuron gets a chemical text message saying, hey, you connected. You're doing a good job. Stay alive.
That's exactly it. It's positive reinforcement. But if the neuron fails to connect or makes a weak connection, it doesn't get that signal. The lack of that signal is interpreted by the cell. It tips the BCL2 balance toward backs and backers, and it quietly undergoes apoptosis.
It's use it or lose it at the most fundamental level.
Precisely.
So vulnerability is the key here. The brain isn't always this sensitive, right? My brain isn't pruning itself on this scale right now.
Not at all. This vulnerability is intensely age-dependent. It is specifically during periods of incredibly rapid growth and critically synaptogenesis, the formation of trillions of new connections.
Which leads us perfectly into Part D, developmental neuroapoptosis. You've flagged this as very high yield. So when exactly is this window of vulnerability in humans?
For the exam, you need to define the brain growth spurt. Miller and Spalting are both very clear on this. This critical window in humans starts from the 3rd trimester of pregnancy.
So even before birth?
Yes, absolutely. It starts in the 3rd trimester and extends through the first few years of life, typically up to about age 3.
So premature babies, neonates, infants, and toddlers are all sitting right in this danger zone.
They are in the absolute peak of the danger zone. This is the period of maximum synaptogenesis. The brain is wiring itself at a furious, almost unbelievable pace.
Okay, and here is the scary part, the exam phrasing. How do we explain how anesthetics might interfere with this delicate process?
This is the core hypothesis, and you need to state it clearly. The theory is that anesthetic agents hijack this natural physiological pruning process.
Hijack it how?
Well, remember, neurons survive if they receive excitatory signals and those neurotrophic factors. That's their stay alive signal.
And anesthetics.
Yep.
They block excitation.
By definition, that's their entire job. That's how they work. They dampen neuronal activity across the brain. The immature neuron interprets this widespread drug-induced silence as a lack of connection.
It thinks it failed the use it or lose it test.
Exactly. It thinks, I'm not connected to anything, I must not be useful, I serve no purpose. And so it commits suicide.
It triggers its own death program.
It initiates the intrinsic apoptotic pathway that we just detailed.
That is a chilling concept. It's not that the drug is a poison in the classic sense, but that it's tricking healthy cells into killing themselves.
Inappropriate death of healthy neurons. That's the phrase to use.
And that leads us directly into Part D, anesthetic-induced neuroapoptosis. This is an exam favorite. Which agents are the culprits?
According to Morgan, Miller, and Stolting, it's a long list. It's not just one class of drug.
That's the worrying part.
It is. For the exam, it's best to categorize them by their mechanism of action.
Okay, let's break down the mechanisms. First up, the NMDA antagonists.
Right. The prototype agent here is, of course, ketamine. And don't forget the gas we use all the time, nitrous oxide, N2O.
How does blocking the NMDA receptor cause cell death?
The NMDA receptor is the main excitatory ion channel receptor in the brain. It responds to the neurotransmitter glutamate. If you block the NMDA receptor with ketamine or nitrous, you are cutting off that critical excitatory input. You are literally removing the survival signal. You're enforcing silence on the neuron.
Okay, makes sense. Mechanism number 2, GABA agonists.
This is a huge category. This covers all the volatile anesthetics, isoflurane, sevoflurane, desflurane. It also covers our main intravenous agent, Propofol, and it includes benzodiazepines and barbiturates.
So basically everything else in our anesthesia trolley.
Almost everything, yes. Potentiating the GABA-A receptor causes widespread inhibition. It enhances chloride influx, hyperpolarizes the neuron, and makes it less likely to fire.
But it also causes silence, just through a different door.
Exactly. And in the immature brain, this profound suppression of activity leads to the downregulation of those survival factors we talked about, triggering apoptosis. But there is a strange twist here that Miller mentions, a paradox.
Oh.
In the very immature brain, we're talking early fetal life GABA, can actually be excitatory, not inhibitory.
Wait, what? GABA is excitatory. I thought it was a universal inhibitor.
It is in the adult brain, but in the very early developing brain, the intracellular chloride concentration is high, so opening a GABA channel actually causes chloride to leave the cell, leading to depolarization. It's an odd bit of developmental neurobiology.
So in that specific, very early window, GABA agonists might cause excitotoxicity death by overexcitation.
That's the theory. But for the main window we worry about in the operating room, the neonate and toddler, the primary concern is the profound suppression of activity leading to apoptosis.
And Miller mentioned something you called a double whammy. What's that?
Yes. This is crucial for your clinical practice and for exam answers. Miller clearly notes that combining an NMDA antagonist like nitrous or ketamine with a GABA agonist like isoflurane or propofol seems to exacerbate the apoptotic damage in animal models.
Which is standard clinical practice of the gas for induction, some sevoflurane for maintenance, maybe some propofol.
Exactly. That is precisely why this topic is so hot and so controversial. Our standard balanced anesthesia techniques could, theoretically, be enhancing this neurotoxic effect.
We've been talking a lot about theory and mechanisms. The examiner is inevitably going to ask for proof. Doctor, show me the data. Let's talk about Part G, experimental and clinical evidence.
You absolutely have to divide this answer into two parts, animal evidence and human evidence. They tell very different stories.
Let's start with the animal data.
The animal evidence is robust, it's reproducible, and it's, frankly, terrifying. You must be able to cite the landmark study by Jevtovic-Todorovic et al. This is mentioned in Miller, Stolting, everywhere.
Okay, tell us about that study. What did they find?
They took 7-day-old rats, which in terms of brain development is roughly equivalent to a human neonate at term. They exposed them to a cocktail of anesthetics, midazolam, nitrous, and isoflurane.
And for how long?
For 6 hours.
That's a very heavy, very long anesthetic for a tiny rat.
It is, and the findings were stark. They found widespread apoptosis throughout the developing brain. But here's the more important part. When these rats grew up, they had significant permanent functional deficits. They couldn't navigate mazes properly. They had clear learning and memory disabilities.
So it wasn't just dead cells under a microscope, it was a dumber rat.
A demonstrably dumber rat, yes.
Okay, but a common criticism is rats are not humans. What about primates? They're much closer to us.
A very important question. Miller cites several key studies by researchers like Slicker and Brambrink in rhesus monkeys. They showed that after 3 hours or more of exposure to clinically relevant doses of either ketamine or isoflurane, there was significant neuronal apoptosis in the monkey brain as well.
That 3-hour mark seems to keep coming up.
It does. It seems to be a threshold in a lot of the animal studies. We'll come back to why that's important for the guidelines.
Okay, so the animal data is strong and points towards a real problem. Now, you have to contrast this with the human relevance. This is where you save your grade. Right. You cannot just tell the examiner that anesthesia kills babies' brain cells.
Absolutely not. That's an instant fail. You have to pivot and discuss the human data with nuance. We have to look at the major clinical trials, both retrospective and prospective. The big three you should mention are PANDA, GAS, and MASK.
Let's unpack those.
Yeah.
What was the PANDA study?
PANDA stood for Pediatric Anesthesia Neurodevelopment Assessment. It was a sibling-controlled study. They looked at kids who had a single, short inguinal hernia repair before the age of 3.
And they compared them to their own brothers and sisters who didn't have surgery? That's a great control group. Same genetics, same home environment.
A very strong study design. And they measured their IQ scores and other neurocognitive functions when they were older, between ages 8 and 15.
And the result?
No difference. No statistically significant difference in IQ or other major outcomes.
Okay, that's one point for it's probably safe.
Right.
What about the GAS trial?
The GAS trial was a big international multi-center randomized controlled trial. They took infants having hernia repair and randomized them to either general anesthesia with sevoflurane or regional anesthesia with an awake spinal.
So they directly compared GA to a technique that avoids GA drugs altogether.
Exactly, the perfect comparison. They then followed these kids and looked for neurodevelopmental outcomes at age 2 and again at age 5.
And the result.
Equivalent outcomes. No difference between the awake spinal group and the general anesthesia group.
So that's two major studies that are very reassuring.
It is very reassuring, but with a critical caveat. Both studies looked at single, brief exposures to anesthesia. The average duration was about an hour.
So there's a caution attached.
There is. The AMRESCA study, which was a birth cohort study from the Mayo Clinic, and other observational studies, suggests that the real risk, if there is one, appears to be linked to multiple exposures or prolonged exposures.
So the kid having one hernia repair is probably fine. The child with a complex congenital condition needing 3, 4, 5 surgeries, each lasting hours. That's the gray area.
That is the population where we still have concern and a lack of clear, reassuring data.
This leads perfectly into Part H factors, influencing neuroapoptosis. If I'm listing risk factors in a short note, what are the key headings?
Number 1 is age. The window of vulnerability is the fetus, the neonate, and the child up to age 3. An anesthetic and a 10-year-old is not the same issue.
Number 2.
Duration. As we saw in the primate data, damage was evident after 3 hours, but not necessarily after shorter periods. Keep it short.
3.
Dose. It is a dose-dependent effect in animal models. Higher AMAC, a volatile agent, equals more apoptosis.
And 4, the one we just mentioned.
Repeated exposure. This is probably the most significant red flag in the human observational data.
And there's a 5th one, which Miller emphasizes heavily, and it's the one we, as clinicians, have the most direct control over in the operating room.
Yes. And arguably the most important, physiological derangements. This is huge. Hypoxia, hypotension, hypoglycemia, severe acidosis, or alkalosis. Miller states very clearly that anesthesia without physiological stability is far, far worse than the drug alone.
So a hypotensive hypoxic brain is going to undergo apoptosis regardless of whether you used sevoflurane or a local anesthetic.
Exactly. Ischemia is an incredibly potent trigger of that intrinsic mitochondrial pathway we drew out. If you let the baby's blood pressure crash or let them get hypoxic, you are causing neuroapoptosis via the ischemic pathway, which is probably far more damaging than any direct pharmacological trigger.
That's a key clinical takeaway. Manage the physiology above all else.
That's your primary job.
Okay, let's move to Part E, clinical implications.
Yeah.
How does all this science change what we actually do on a Monday morning?
So Part 9, pediatric anesthesia. The examiner puts you in a scenario. Doctor, you're in the pre-op clinic. The parents of a 1-year-old are worried. They read about brain damage on Google. They ask you if you should cancel the surgery. What do you say?
What's the answer?
You have to talk about balancing risk. Morgan and Miller are very clear on this. You do not withhold anesthesia for necessary surgery.
Why not? Why not just, operate with them awake or have people hold them down like in the old days?
Because the physiological stress response to that is catastrophic. Uncontrolled pain causes a massive catecholamine release, hypertension, tachycardia. This in itself causes excitotoxicity and is damaging to the developing brain. Operating on a conscious, terrified baby is not an option. It's unethical and it's physiological suicide.
So the stress of surgery is worse than the risk of the anesthetic?
For necessary surgery, yes.
Okay, but what about elective surgery?
That's a different calculation. The consensus, which is reflected in the FDA warning that Stulting discusses, is to consider delaying purely elective procedures until after age 3 if it is medically safe to do so.
So something like a cosmetic ear correction or removing an extra digit.
Wait until they are older, but a hernia that is at high risk of incarceration, you do it now. A complex congenital cardiac defect, you have to do it now. The risk of the condition itself far outweighs the theoretical apoptotic risk.
Right. It's life and limb over a theoretical risk to neurodevelopment.
You have to be pragmatic. If they don't survive their heart defect, their future exam scores don't matter.
A stark but accurate way to put it. Now, Part 10 covers the ICU and critical care. This isn't just about elective surgery in healthy kids.
No, not at all. Miller and any good ICU manual will highlight that apoptosis is a major mechanism of secondary brain injury and many other conditions we see.
Like what?
In stroke, or after cardiac arrest, it's the ischemia and reperfusion injury that drives the intrinsic mitochondrial pathway. In sepsis, the storm of inflammatory cytokines, particularly TNF-alpha, directly triggers the extrinsic death receptor pathway.
And traumatic brain injury, TBI.
PBI is a classic example. You have the primary injury, which is the physical smashing of cells, that's necrosis. But then, for hours and days afterwards, there's a wave of secondary injury that spreads through the surrounding salvageable penumbra tissue. And that secondary death is largely apoptotic.
So our neuroprotection strategies in the ICU, like therapeutic hypothermia or avoiding hyperglycemia, controlling seizures, are they partly trying to stop this?
They are absolutely aiming to block these apoptotic cascades to save those penumbral cells. That's a huge part of neurocritical care.
This brings us to Part F, prevention and safe practice. The examiner asks, okay, doctor, so how do you anesthetize a 6-month-old safely, given all these risks? You need a concrete strategy.
You need a simple 3-point plan. First, avoidance. Is this anesthetic truly necessary right now? Can the surgery wait? If it's purely elective, the safest anesthetic is the one you don't give. Defer it.
Okay, number 2.
Optimization. This is the most important practical point for the clinician. Miller says, a physiological anesthetic is a safe anesthetic. You must maintain blood pressure, oxygenation, normocarbia, and normoglycemia within tight limits. Don't let them get cold. Don't let them get hypotensive.
And third,
duration. Keep the anesthetic and surgical time as short as is safely possible. This requires a team approach. Efficient surgery matters. Don't waste time after induction. Be ready to go.
what about the role of multimodal anesthesia, using regional techniques?
Yes, that fits in perfectly. Use regional anesthesia spinals, caudals, nerve blocks, whenever it is appropriate and possible. This allows you to significantly reduce the total dose of the systemic general anesthetic agents.
Are local anesthetics completely free of this risk?
Miller notes that while local anesthetics have their own potential toxicities, like AST, they don't generally cause this central neuroapoptosis mechanism we've been discussing. So yes, a good regional block is a cornerstone of a neuroprotective anesthetic.
What about using opioids to spare the volatile agent?
Yes, that's also a valid technique, but remember, opioids are often necessary anyway to blunt the surgical stress response, which is also neurotoxic. It's always a balance. Don't underdose pain relief just because you're scared of a volatile agent. An awake, screaming baby on the ventilator is also bad for the brain.
Right. Let's discuss the guidelines in Part 12. You mentioned the FDA warning a few times. Let's get specific on that.
The FDA Drug Safety Communication, issued in 2016, is a critical document to be able to cite in an exam. It specifically warns that repeated or prolonged use of general anesthetic and sedation drugs during surgeries or procedures in children younger than 3 years or in pregnant women during their 3rd trimester may affect the development of children's brains.
And prolonged was generally defined as?
Generally greater than 3 hours, based on the primate data.
So what is the balanced conclusion that Miller suggests you should give to an examiner?
The conclusion is one of careful reassurance, not alarmism. You say that while the animal data is strong and biologically plausible, the current human evidence is reassuring for the vast majority of cases we do, which are single, brief anesthetics for necessary procedures.
So we shouldn't be alarming parents unnecessarily.
Correct. But we should remain cautious and vigilant with cases involving prolonged or repeated exposures. And our main focus always must be on maintaining meticulous physiological stability. That is our best neuroprotective strategy.
Okay, we are in the home stretch now. Part G, exam integration. Let's visualize some tables and charts for the listeners. This is how you structure your written answer to make it easy for the examiner to give you marks.
Absolutely. Section 13, comparison tables. You should physically draw these.
Okay, let's verbalize table 1. Apoptosis versus necrosis.
4 rows. First row, cause. Physiologic or pathologic for apoptosis versus always pathologic for necrosis. Second, cell size. Shrinkage for apoptosis, swelling for necrosis. Third, membrane. Intact with blebbing for apoptosis, disrupted for necrosis. And fourth, the big one, inflammation. No for apoptosis, yes for necrosis.
Clear, simple, easy marks.
Okay.
Table 2. Physiologic versus pathologic neuroapoptosis.
Even simpler. Physiologic. The purpose is embryogenesis, synaptic pruning, sculpting the brain. Pathologic. The cause is drug-induced, ischemia, sepsis, or traumatic brain injury. If you put those 2 tables in your answer, the examiner immediately sees clarity of thought.
Now, section 14. Viva traps. This is critical. I'm going to play the examiner. You are the candidate. I'm staring at you over my glasses.
Candidate, is Propofol safe for a 2-year-old?
Okay, this is a classic trap. If you just say yes, you sound ignorant of the basic science. If you say no, you sound clinically naive because we use it every day.
So what's the safe answer? The answer is, sir, Propofol is a potent GABA agonist. In animal models like other GABA agonists, it has been shown to cause neuroapoptosis in the developing brain. However, in human clinical practice for short, necessary procedures, the evidence from major trials like PANDA and GAS suggests it is safe with regard to long-term neurodevelopmental outcomes. I would use it judiciously, while ensuring meticulous hemodynamic stability and avoiding any prolonged or unnecessary exposure.
That is a perfect answer. You acknowledged the science, you quoted the clinical evidence, and you brought it back to safe practice. You didn't say, yes, it's 100% safe, and you didn't say, no, it kills brains. Nuance is your friend.
Always.
Next trap. What about ketamine? It maintains respiration, it keeps the blood pressure up. Surely it's a better choice.
Sir, while ketamine has an excellent hemodynamic profile, it is a potent NMDA antagonist. Mechanistically, it is also strongly implicated in causing neuroapoptosis in developing brains in animal models by blocking essential excitatory survival signals. Therefore, from a neurotoxicity perspective, it is not considered to be devoid of risk.
Okay, final trap. So should we just do a spinal on every child instead of a general anesthetic?
Sir, regional anesthesia is an excellent component of a multimodal strategy to reduce systemic drug exposure and is a great alternative in the right circumstances, as shown in the GAS trial. However, performing a successful spinal in a very young child requires specific expertise and can be challenging. It also often requires some sedation to keep the child still, which reintroduces the risk of sedative drugs. The decision between regional and general anesthesia must be based on the specific surgical needs, the patient's condition, and the skills available in the department.
Excellent. You've shown balanced, practical judgment. Now, section 15, diagrams. If I have to draw something on the paper, what's the highest yield thing to draw?
Draw the intrinsic pathway flowchart. It takes 10 seconds. Start with a box at the top, cell stress anesthetic agents. Draw an arrow 2. Mitochondria. From the mitochondria, draw an arrow out labeled Cytochrome C release. Arrow from that to Apoptosome formation with APA1. Arrow 2, activation of Caspase 9. Arrow 2, activation of Caspase 3. And a final box at the bottom, Apoptosis.
Simple, linear, effective.
That sketch alone could be worth half the marks for a short note.
And maybe the vulnerability timeline.
Yes, a simple graph. The x-axis is age from 3rd trimester to, say, 5 years. The y-axis is vulnerability to neuroapoptosis. You draw a curve that starts high in the 3rd trimester, peaks around birth and the first year during peak synaptogenesis, and then tapers off significantly by age 3.
Okay, let's wrap this entire deep dive up with the summary and exam conclusion. We need the elevator pitch for the examiner. If you had the 30 seconds to summarize the topic, what would you say?
If you're summarizing this topic in a viva or at the end of a long answer, you must hit these 6 points. I'll start, you finish. 1. Definition. Apoptosis is programmed, energy-dependent cell death, and it's fundamentally distinct from necrosis.
2. Physiology. It's a vital and necessary process for normal brain development, responsible for synaptic pruning.
3. Pathology. Our anesthetic agents, specifically NMDA antagonists and GABA agonists, accelerate this process in animal models. You can cite Miller here.
4. Evidence. The human evidence for a single brief exposure is reassuring. You must mention the PANDA and GAS trials.
5. Guidelines. The FDA warns against prolonged, which is more than 3 hours, or repeated use in children under the age of 3.
And 6. Practice. Our safe practice focuses on meticulous optimization of physiology, avoiding hypoxia and hypotension, and avoiding unnecessary procedures in this age group.
That's it. If you can recite those 6 points, you've mastered the topic.
That is a solid, unbeatable framework. Whether it's a short note, a long answer, or a viva, if you stick to that structure, you will pass.
You'll do better than pass. You will demonstrate that you understand both the deep science and the pragmatic clinical reality, which is what separates a good candidate from a great one.
Amazing. To our listeners, especially you, preparing for the Gujarat University exams, good luck. You have the sources, you have the structure, go crush it.
Absolutely. Read the question carefully, keep your definitions precise, and don't panic. You've got this. Good luck.
Deep dive. Complete.